Okay, let's jump right into what might be one of the most clever, truly counterintuitive ideas in cancer therapy in a long time. It's a story about taking one of our body's most violent, most aggressive defense mechanisms and turning it from a massive problem into a life-saving weapon. And it all comes down to a really, really smart disguise. So let me just ask you this. What if you could trick your body's most violent defense mechanism into attacking your own cancer? What if you could paint a giant flashing target on a tumor, a target so obvious, so completely foreign, that your immune system, which, by the way, is already armed and ready to go, would just launch an all-out, no-holds-barred assault against it? We're not talking about teaching it something new. We're talking about unleashing a power it already has. So researchers have this really cool nickname for this approach, the Circe Strategy. Now, if you remember your Greek mythology, Circe was the sorceress from the Odyssey who, turned Odysseus's men into pigs. And believe it or not, that is exactly the goal here. It's a bit of biological sorcery to make cancer cells look to our immune system just like pig cells. And the results? Well, honestly, they're just stunning. Okay, so to really get how this new weapon works, we've got to first understand a really old, really powerful enemy. I'm talking about a biological force that's been a major roadblock for medical progress for decades. This enemy, it's called hyperacute rejection. And for years, this has been the #1 reason we can't just use pig organs to save human lives. You see, when a pig organ gets transplanted into a person, the immune system doesn't just, attack it. It annihilates it. We're talking minutes to hours and it's gone. It is genuinely one of the most violent, explosive reactions in all of biology. And the craziest part, the trigger for all this violence is shockingly simple. It really all boils down to this one tiny sugar molecule called alpha-gal. See, pig cells are just covered in this stuff. Our human cells, not a single one. So our immune system, over millennia, has basically learned to see this one little sugar as the ultimate danger signal, a universal sign of a foreign invader that must be destroyed on sight. And this is the crucial point. Our bodies are already locked and loaded. We don't need any special training. Right now, every single one of us is walking around with a whole arsenal of powerful antibodies just waiting, just itching to attack this specific pig sugar. The weapon is already there. It's built in. It just needs a target. And that is the absolute genius of this whole idea. Okay, so the weapon is there, right? The antibodies are ready to go. But how in the world do you actually put this disguise onto a cancer cell deep inside the body? Well, you're gonna need a Trojan horse. And here it is. It's a brilliantly engineered virus called NDV-GT. Let's break it down. The horse part, the delivery vehicle, is something called Newcastle disease virus. Now, that's an avian virus, a bird virus, which is totally harmless to us, and super important, our immune systems have never seen it before. So it's basically invisible. It can sneak right past our defenses. And the soldiers hidden inside that horse? It's the payload, the one single gene that gives the cell the instructions to make that pig sugar. So here's how this all plays out. It's a beautiful 4-step process. Step 1. The virus finds and infects a cancer cell. Simple enough. Step 2. It drops off its payload, that gene, and forces the cancer cell to start producing that pig sugar all over its surface. It puts on the disguise. Step 3. Bam. Our pre-existing antibodies, the ones that have been waiting around, spot this pig cell, and they trigger that insane hyperacute rejection. But here's the key. It's not happening to a whole organ. It's happening right inside the tumor. And finally, step 4, the attack is so violent, the chaos is so massive, that it not only kills the disguised cancer cells, but it sends out all these alarm signals that basically wake up and train the rest of the immune system to start hunting down all the other cancer cells, even the ones without the disguise. It's a chain reaction. Okay, that's a brilliant theory, right? It's elegant, it's clever, but the billion-dollar question is always the same. Does it actually work in real people, in patients? Well, let's take a look at the verdict from the very first human study. 90%. Just let that number sink in for a second. 90%. We're talking about a trial with patients who had very advanced cancers. These were people who had run out of all other options. And in that group, this therapy got a 90% disease control rate. Think about that. 9 out of 10 people whose cancers were actively and aggressively growing saw their disease either shrink or just stop dead in its tracks. So, what does disease control actually mean here? Let's break it down. For 5%, one patient in this trial, their cancer completely disappeared, a complete response. For another 30%, their tumors shrunk significantly, and for a whopping 55%, over half the patients, their tumors just stopped growing. They became stable. And you have to understand, for people in this situation who have been told there's nothing left to try, just hitting the pause button on your cancer is an absolutely monumental victory. And this table gives you the whole picture. I mean, look at the variety of cancers they treated. Liver, cervical, lung, breast, all sorts of really tough, advanced solid tumors. But for me, the most stunning number on this whole slide might be that last one. 0. 0 serious adverse events. You guys, getting this level of effectiveness with that kind of safety profile is almost unheard of in cancer treatment. It tells you the attack was incredibly powerful, but also laser-focused, contained right where it needed to be, inside the tumor. And to put a real human face on all this data, just listen to this quote from the trial report. That patient, the one with cervical cancer who had a complete response, the report doesn't just say she's in remission, she was described as being clinically cured. Cured. That's the kind of life-changing, transformative impact we're talking about here. Okay, so let's pull back and look at the bigger picture for a minute. These results on their own are incredible, but the real potential here, it might be even bigger. We might not just be looking at a new key for a few locks. This could be a master key that unlocks the full power of a whole bunch of other cancer treatments. So probably the biggest challenge in immunotherapy today is this problem of so-called cold tumors. You can think of them like stealth bombers. They are essentially invisible to the immune system. And our most powerful modern drugs, things you've heard of like checkpoint inhibitors, they just don't work on them. I mean, it makes sense, right? If your immune system can't even see the tumor, you can't really encourage it to attack it. Well, this is where NDV-GT completely changes the game. It solves that cold tumor problem by basically setting off a 5-alarm fire right in the middle of that stealthy, invisible tumor. That hyperacute rejection we talked about, the cascade it sets off is so massive, so incredibly inflammatory that it's like setting off a giant flare in the dark. It makes the tumor absolutely impossible for the immune system to ignore. In other words, it turns cold tumors red hot. And what this does is create this perfect, powerful combination with drugs we already have. The best analogy is a car. Think of the NDV-GT therapy as stepping on the gas. It creates a powerful new T-cell response from scratch. It gets the engine roaring. Then you can bring in the checkpoint inhibitors, and they do what they do best. They stop the brakes. They stop the cancer from shutting down that attack, letting that engine redline and just keep going. And it doesn't stop there. This could also solve the biggest problems facing another revolutionary therapy you've probably heard of, CAR T. Now, CAR T cells are these amazing living drugs that have worked miracles for blood cancers, right? But they have just consistently failed against solid tumors, and it's basically for 3 big reasons. 1, they have a hard time finding a good universal target on the tumor cells. 2, they can't physically get inside the solid tumor. And 3, even if they do get in, the tumor's environment is so hostile, it just deactivates them. Well, guess what? That 5-alarm fire that NDV-GT starts, it helps solve all 3 of those problems. It creates a universal target, that pig sugar. The massive inflammation breaks down the tumor's physical walls so the T cells can finally get in. And it completely rewires that hostile environment to be welcoming to the attack. This could seriously be the breakthrough that finally paves the way for CAR T to work against solid tumors. So this all brings us back to that final, really provocative question. By taking a biological barrier and turning it into a weapon, and by taking invisible cold tumors and turning them into unmissable red-hot targets, have scientists actually found the master key? Is this the one therapy that not only works incredibly well on its own, but also unlocks the full, true potential of our entire arsenal of cancer immunotherapies? It's early days for sure, but the first signs, they're suggesting the answer might just be yes.